All right, so in this episode, what we're going to be doing is we're going to be touching on whether things that are known as cladograms, which are these phylogenetic tree hypotheses, we're going to be taking a look at whether or not those are falsifiable in the Popperian sense, right? This episode is going to be somewhat more heavy on philosophy and logic, and it will take a look at who Karl Popper was and also his criteria of falsifiability, which is essentially the idea that scientific theories must be testable in some way in order to potentially be refuted by the evidence. So I have touched on some of these points, at least when I was going over universal common descent and whether or not universal common descent could be falsifiable. And so we talked about these auxiliary hypotheses. In this episode, at least, we're going to be touching on a lot of those similar concepts, but specifically, we're going to be taking a look at cladograms and as well as systematics, which is the field that involves these phylogenetic trees. We're basically going to be showing that these cladograms, and we'll take a look at some of the examples of these cladograms, but we'll show that they're not falsifiable in principle, which means that there's no conceivable observation that could conclusively prove a cladogram wrong. So we're also going to bring this in light of Popperian philosophy and look at the way Popper frames his whole falsifiability principle. Okay, so what is Popperian falsification? So essentially, falsifiability is the idea that a hypothesis makes a testable prediction which could, at least in theory, conflict with some possible observation. So a falsifiable hypothesis forbids certain outcomes, and if those outcomes do occur, then the hypothesis would be proven false. For example, saying statements like all swans are white, at least in theory, that is falsifiable because finding a black swan would obviously refute it. So in Popper's view, falsifiability is what demarcates or what kind of separates science from non-science, right? So to call something actually science, it has to be falsifiable. Otherwise, it's just pseudoscience. And to be like more clear, Popper's falsification is essentially the view that science advances by making these bold claims, and these bold claims are tested by attempts to refute them. Basically, according to this hypothesis of falsifiability, a hypothesis should rule out some possible observations in order for it to be corroborated. And so corroborated here means the hypothesis is supported by surviving these rigorous kinds of tests. With that in mind, we can also introduce what cladograms and phylogeny are. So cladograms are basically a branching diagram, and I have an example of this on the next slide, where it basically shows a hypothesized evolutionary relationship, like what organism is more closely related to what other organism. So it's essentially somewhat of a phylogenetic tree that is derived through data of a particular trait or of a particular organism. And that what that tree does is it forms these branch points that represent common ancestors. Unlike a family tree, a cladogram usually does not indicate the exact time or ancestry, but rather it kind of groups these shared traits or these taxa through what's known as synapomorphies. So this is an example. Anyone who's taken an introductory course to biology would know how to read cladograms and what they are. But essentially, the idea here is that you have a number of these evolutionary relationships between organisms, and what kind of groups them into a taxa is these shared characteristics. For example, crocodiles and eggs have the shared characteristic of eggs in shells, right? And so, for example, primates and rabbits have this shared characteristic of possessing hair or fur. And you can drive these trait characteristics. So the further back you go, you can see how they're related. So for example, sharks are related to everything else in this diagram through this particular trait, which is particularly that they're all vertebrates. Essentially, these red lines here, these are the shared traits, or you can call them synapomorphies. And I'll explain that more in the next slide. But basically, what we'll be showing in this presentation is that this picture right here is not really falsifiable, at least in principle. Basically, there are no possible observations of present day traits that could logically prohibit any given cladogram. So, like, why is this cladogram better than any other cladogram, at least in theory, right? Something that doesn't rely on parsimony. And we will get to all these things later on in the presentation. But what is it with regards to this cladogram that proves this particular cladogram? Again, this is supposed to be a hypothesis, right? So how do we prove it? And essentially, we'll show that there's absolutely no data that you can give that would contradict this particular diagram. You can always kind of invoke a one-off event or somewhat of the homoplasius independent trait that is not synapomorphic. And what that means is that essentially that shows that you can rearrange the tree in whatever way you want to arrange it, irrespective of the data, which is why these are not falsifiable. This is important because cladograms are quite a staple in evolutionary biology. Everyone uses them. Everyone tries to model their relationships of organisms based off of these systematic cladograms, and they attempt to show that this is a good way to orient the tree of life, for example. But again, this is important to realize that it's not really falsifiable. And if that doesn't make any sense, it's okay because in the next couple of slides, I'll be going over this in more detail. Why cladograms aren't really falsifiable. Now, cladograms, they can't really be falsifiable because there's no observation that could falsify them. This is going back to what I said just a moment ago. So the concepts of cladograms are basically too accommodating in the sense that they can fit any unexpected observation. Actually, on top of my head, I remember in 1997, I believe was the date, there was a researcher named Klug, and what he tried doing was he tried to argue that you can build these cladograms based off of synapomorphies. And again, these synapomorphies are these particular traits. So like hair, eggs, and shells. So he's basically saying that you can build, since we build these cladograms off of these synapomorphic traits, he says that we can also falsify them off of these synapomorphic traits as well. Again, what are synapomorphies? Just to explain it in better words, a synapomorphy is basically a shared kind of derived character or like a trait that's present in a particular ancestor and all of its descendants, right? And so therefore that defines a clade. So in this example, crocodiles and birds are a clade, and the ancestor has eggs and shells. Again, this shows that this is a particular synapomorphic trait that's shared by both crocodiles and birds. Another example that's not really shown on this particular tree, but it's always mentioned in the books, is a synapomorphic trait would be something like feathers. So, for example, feathers on birds are pretty much synapomorphic traits because all birds have these feathers and their common ancestors did as well. So something like, for example, reptiles did not have these feathers, therefore they would not be classified in the same clade as those birds. Now, the issue is that is this really a way to falsify cladograms? Can we use synapomorphic traits to falsify a cladogram? And the issue is that Klug himself admits this. So Klug was the one who brought up this distinction. We can falsify cladograms through synapomorphic traits, but he himself also concedes. He says that a cladogram is logically consistent with all synapomorphy distributions, congruent or incongruent. And so that's again really important because that was going back to what I said. So why can't synapomorphic traits disprove a cladogram? That's because you can rearrange the synapomorphic events in any particular way you want and still end up with this tree. And that's the whole point, is that a cladogram will always be logically consistent regardless of how you distribute those synapomorphic traits. That is why it's not falsifiable. And to test the hypothesis implies that we can find, at least in principle, we could find some observation out there that could disprove this cladogram. But what we're looking at is the fact that because these cladograms are compatible with any specific synapomorphic trait or any distribution of a trait, then that implies that these are not really testable because it's always going to be logically consistent with whatever distribution that we come up with. No matter what pattern of traits that we observe in that taxa, so even traits that conflict with the tree, you can always imagine some scenario that fits those observations into that tree. This means that cladograms cannot be logically contradicted by any particular data. So again, just to flush this out in more clear terms, because I feel like this is somewhat of a complicated topic, but ideally, if we look at how we test something, we have certain assumptions. So we have something called a background assumption. In this case, if we're looking at a cladogram, the background assumption would be something like descent with modification or like evolution, for example. That is our background assumption. Now, we also have a specific tree hypothesis. So we're actually looking at a hypothesis. This is in particular a given hypothesis of how sharks are related to fish and to amphibians and to primates, etc. So what we're saying is that we have two things in our mind. We have number one, the specific hypothesis that we're looking at, in this case, the cladogram or the tree, and also this background assumption of evolution. Now, what we're saying is that there are no specific tree hypotheses, right, that could falsify, or there's no specific data that could falsify this specific hypothesis. What I'm saying is that essentially, whatever trait that we could involve, whatever synaptomorphic event that we can have, if it doesn't fit into the data, then we can just in terms of making it somewhat of an ad hoc explanation, we can redistribute that particular trait elsewhere, and we can do that under the guise of, for example, homoplasia or independent evolution. Let's say, for example, there was, and I'll give an example later in the slides, but let's say there was a specific gene that was similar between amphibians and sharks. So far between amphibians and sharks, we say that they're related in this particular manner because they both are vertebrates, right? But then the question is, why aren't amphibians more closely related to sharks than the ray fin fish are to sharks? What's the relationship? And organisms like these that clearly look different and sound different and might even behave differently, it's somewhat obvious. But once it becomes a matter of species within even the same phyla, then that's when, this dilemma shines forth. But essentially, what I'm saying here is that why isn't it like, let's say there's another hypothesis, another tree where amphibians were more closely related to sharks than to fish? How would that make sense? How would we disprove one tree over the other tree? Let's say hypothetically there was a specific gene that amphibians shared with sharks. So what we can do is we can say, look, because there's this gene that amphibians share with sharks, therefore we should prefer a diagram that does not have these ray-finned fish in between these amphibians. So essentially what I'm saying is we should, let's say there's a tree that did not have this, and instead it just linked these two were closely related. How would we know if that tree is better than this tree right here? And what I'm saying is that we can look at certain genetic synaptomorphic events or synaptomorphic traits because that's what the proposed theory or proposed solution is, right? We can falsify a particular diagram based off of these synaptomorphic events. So if that is true, then let's say we find in particular a trait that amphibians have and sharks have, because again, it's shared by amphibians and by sharks. That's good, strong evidence. But does that disprove this particular tree? Does that actually disprove anything? And the answer is no. And it's no because instead of putting it here, you can also individually put them here. So what does that mean? So let's say these lines that I have just put in, so like this line and this line, we're saying that these are synaptomorphic traits, but we're saying although they're the same trait, they're not shared between the sharks and the amphibians. If they were shared, they would have come over here. This is how we show a homoplasious trait. So we say that they're independently evolved. And because they are independently evolved, and because we can redistribute these traits in whatever way that makes sense, what that means is that at least in theory, these cladograms cannot be falsifiable. Again, we're going to be looking at proposed solutions that argue, you know what, like maybe the best one's the one with the least or the most parsimonious, for example, or the one with the least homoplasious, like ad hoc assumptions. And we will look at whether or not those are viable solutions to Popper's falsification. But the point I'm bringing forth right here is that pretty much there's no observation that we can give that would falsify this particular diagram. There will always be the ability to redistribute the trait in an ad hoc way. And again, there's two possibilities. Either that trait that I just drew, either it's based off of homology. So again, homology means that it's inferred from the same common ancestor, in which case it would be right here if it's homology. If that trait was not homologous, it would be homoplasious, and we would say that they are independently distributed. And I hope you see the point that I'm trying to put across here, which is that a given cladogram can truly accommodate any scenario that you give it. You can always redistribute the specific event. So that's why you can't really falsify them based off of these synaptomorphic traits, because there's always a scenario that can redistribute that trait and still preserve the hypothesis. I think to conclude on this point, before I go on to the next point, the idea is that hypotheses of these phylogenetic relationships or these clades, again, they're not falsifiable. We can't really deduce some kind of test that could falsify them. But there is a very important point that I should bring up here, which is that just because it's not falsifiable does not mean that's not fallible. And that's a huge thing that I need to bring up here. Essentially, I'm saying is that in Popper's framework, and again, we will be going over Popper's framework in more detail, but there is a difference between something being fallible, in other words, something could be wrong, and something being falsifiable. And so, what I'm trying to say here is that it could be the case that this tree is in fact fallible in the sense that it could be false, but we just have no deductive test to prove that it's false. And that's essentially the major point, which is that cladograms, it's not, the question is not whether or not it's false. The question is whether or not they are falsifiable. And that's a huge caveat and a huge distinction that needs to be made. So, in general, all I'm saying at the end of the day here is that in Popper's framework, because cladograms can't be falsified, they also can't accumulate corroboration. So, we say that a hypothesis is corroborated once we show that the hypothesis has survived different testing methods. But we just said that there is no particular method that we can do, or testing method that we can use to falsify a particular tree. Now, what that means is that because we can't falsify that tree, that means that no particular trait could be corroborated in kind of a phylogenetic tree because there's always a potential to redistribute it, meaning there's always a potential to hand wave away that particular evidence. And so, because it's not really falsifiable, it's not really corroborated. And based off of this, I hope you see what Popper's demarcation principle really is, which is that cladograms, like, they can't really count as scientific because of that particular demarcation principle. So as an example, I did just go over an example, but I can go over one more time. So let's say that we have a cladogram that kind of groups something like whales and deers. Let's draw something like this, right? Now, we say that this right here are whales and this right here are deers. And then let's say something like this right here would be a fish. Now, this is not really an accurate cladogram, and it's not accurate because this is not really telling much in the sense that there's a lot of detail that's omitted from this cladogram. But for the point of the example, I think it should do. So what I'm saying here is that suppose that we have a cladogram that kind of groups these whales, deers, and fish together. Now, if we discover a whale, so let's say there's this particular whale that has a particular gene sequence that is very close in similarity to fish. So let's say hypothetically, we find a gene that a whale has that the fish also has. Let's say hypothetically it's a completely identical gene. Now, would that falsify this particular diagram? Because what is this diagram showing us? This diagram is showing us that whales and deers are more closely related than whales and fish, because whales and deers form one clade, while the fish are somewhat of a more distant relative. So what I'm saying here is, okay, let's make it a bit more detailed. So let's say that whales and deers are closely related primarily because they have a particular trait, whatever synapomorphic event that is. So I'm putting it right here. So whales and deers, let's say whales and deers are similar because they share trait X, for example. So what we're showing here is that whales and deers are common relatives or are close relatives because they share X. Now, hypothetically, let's redraw this diagram a different way. So this is one hypothesis. All right, so again, this is one particular hypothesis. This shows us that whales and deers are close related and fish are a distant relative. Now let's redraw this a cladogram with a different hypothesis. Let's see if I can move this. And in hypothesis two, let's say we have, in this case, deers are going to be here, And we're going to say these are going to be whales, and these are going to be fish. Let's just look at these two hypotheses. So this is hypothesis one, and this is hypothesis two. Now, the question is, what observation would prove hypothesis one over hypothesis two? And again, prove in the sense of which one would falsify hypothesis one, or which observation would falsify hypothesis two. So what we've shown in hypothesis one is that whales and deers share this common characteristic X. That's why they're closely related to deers as opposed to fish. Now, on the other hand, let's say, again, same thing, they share X, but suppose that we found another gene. Suppose that we've looked at another gene, and that gene was, let's say, Y. And gene Y, okay, sorry. Let's say we found a gene Y, right? Now, gene Y says that it's shared between whales and fish. So whales have it as well as fish have it. So what we showed was at first there was this gene X that's shared between whales and deers, but now we found gene Y that's only shared between whales and fish. So what we'll do is we'll draw that here. And in this case, it would look right like this. In this case, this is Y, and again, it's Y and Y. So we have a particular trait, which is Y. Now, what we've shown was that is the occurrence of Y, or the finding of Y, that specific gene that's shared between fish and whales, did that falsify anything? And what we showed is no, it actually didn't falsify anything, because in both cases, this would be X and this would be X. So in both cases, both the whales and deers are sharing something that, even though they're not closely related, and in this case, the whales and fish are sharing something that's not closely related. But the idea here is that regardless of the actual X and Y distribution patterns, every single cladogram is logically consistent with whatever distribution trait that we're looking at. If you notice here, there's two X's and one Y, and there's two Y's and one X. And that shows that regardless of the observation that we find, we can just invoke a homoplasius explanation and redistribute the specific organisms on this tree, sorry, the specific distribution trait of those synapomorphic events. Which, what that means is that our theory could survive anything. That's quite telling. So I hope that kind of makes sense here, which is that whatever lineage we're looking at, both hypotheses are consistent with these particular distribution traits. So to move on, now here's where we're going to go into Popper's methodology and what Popperian falsification involves, and how do we actually formalize this falsification principle. For anyone who's actually read Popper, you would know that what Popper is basically doing is he's reacting to Hume's problem of induction. And so he's basically insisting that science should rely on what's known as deductive logic, which is pretty much reasoning from general hypotheses to specific expected observations. And so induction is basically the idea of generalizing something from observations. And what he says is that induction can never fully be justified. So instead of proving a hypothesis, we should test bold predictions deduced from it in attempts to falsify that particular hypothesis. And so this gives rise to what's known as Popper's hypothetical-deductive model. And the hypothetical-deductive model of testing is essentially this. It's basically saying that we start with some hypothesis H. And so we start with hypothesis H, and we couple that with some background knowledge. And so what we do is once we couple the hypothesis and some accepted background knowledge, from there we can deduce a predictable observation. So we are basically saying that H and B, or H plus B, whatever, however you want to say it, it's going to give us a particular observation E. So in the literature, observations are E. So to repeat, Popper's hypothetical deductive model entails that there's some hypothesis H, and it's coupled with background knowledge B, and it predicts some observable outcome E, right? Now, basically, the idea here is that E should not happen. So if E does occur, then that would falsify the hypothesis. It could also falsify the background as well. We just don't know. And I'll touch on this in a bit more detail in a second. For H and B, we deduce that a prediction of a observable outcome E should not occur if H and B is in fact true. So when we look at the real world, if we do observe this kind of forbidden event E, for example, then logically the conjunction H and B must be false. So this is basically the way that modus tollens works. So the logic behind modus tollens is essentially if H and B implies not E, but in fact E does occur, then H and B is falsified. So for anyone who's studied logic, this is how modus tollens works. It's pretty much how Popper is formulating his criteria for how to falsify a particular or how to test if something is falsifiable. Again, it's important that the hypothesis has to forbid something apart from our background knowledge. Because if no observation could contradict that hypothesis, then that hypothesis isn't really falsifiable. Essentially, what I'm saying here is that if the hypothesis is making no predictions, so if this hypothesis is not making a prediction at all, if it's not saying that there's a non-E, then it's not a real hypothesis. It's not really a testable hypothesis. Now you're doing pseudoscience. You're not actually doing science, right? Because it's not really falsifiable. And again, it's if no observation could contradict this H, if nothing can contradict the hypothesis, then it's not falsifiable. A hypothesis is kind of the hypothesis's empirical content. It has to be equated with somewhat of a range of possible observations that it forbids. And so the more that a hypothesis forbids, the riskier and the more strong the hypothesis becomes. And so, just as an example, let's take a highly falsifiable hypothesis. A highly falsifiable hypothesis makes really bold claims. So something like no mammals lay eggs. Now, that's clearly a very bold claim. And it's bold because finding even a single egg-laying mammal would completely refute it. If we test that hypothesis and we look everywhere and all mammals don't lay eggs, for example, then the hypothesis is very corroborated and it's actually a very strong hypothesis. Again, it's a strong hypothesis because it's making a bold claim, and so far no evidence has disproven the bold claim. Now, in this case, obviously it's false because mammals do lay eggs. Or in this case, like no mammals laying eggs would be easily falsifiable if a single mammal would lay an egg. That would clearly refute it. But then you can also, as an example of a pseudo hypothesis, let's take, for example, a non-falsifiable one. So, for example, all organisms will either have live births or eggs. And so, if you reformulate the argument like that, when you say that organisms will either have live births or eggs, then now you're not really risking anything anymore because you're not explaining anything new. Obviously, if it's an organism and it's reproducing, it's either going to be through some type of birth or it's going to be through some type of egg laying process. And now you're not really making anything that's falsifiable, meaning there is no observation out there that could falsify that hypothesis. And so that is, in particular, that's an example of pseudoscience where a particular hypothesis is not proven false through any specific observable outcome. So in other words, that hypothesis doesn't predict anything falsifiable. Now, this brings up a particular issue that's known as the Duhem-Quine thesis. So I just said that when a thesis fails, it fails because H and B is not true. In other words, if H and B, if it is true and it leads to non-E, for example, that's the hypothesis, the prediction that there won't be an E, and we do end up finding E, so E does occur, then that means that H and B, which is hypothesis and background, is false. So H and B is false. But what is this Duhem-Quine thesis? What is this all about? It's basically saying that since we found E, what is at fault here? Is it H that's at fault? So is the hypothesis wrong? Or is the background wrong? Which part is wrong? It's really hard to isolate which part is actually wrong. So if a test yields a result that shouldn't happen, and by shouldn't, it contradicts the hypothesis, we only know that the combination of H and B is inconsistent. In other words, we only know that the combination of hypothesis and the background knowledge is inconsistent. But perhaps the hypothesis is wrong, or it could even be that one of the background auxiliary assumptions is also wrong. And so we saw this in the previous episode, I think it was episode 3, where we tried to test universal common descent. And what we said was when we take UCD, right, universal common descent, we couple it with some background knowledge, right? So let's take some background knowledge. And this is a refresher of the third episode, right? And what we said is this should make a predictable outcome, right? So some X. But when X does not occur, what bites the bullet? Does UCD end up biting the bullet, or does the auxiliary hypothesis or the auxiliary assumption end up biting the bullet? And what we showed in those episodes was that what ends up biting the bullet is the background knowledge, right? So that ends up being disqualified, not UCD, not the hypothesis, right? But usually, the way that science is done is we want to isolate the hypothesis, right? We want to falsify the hypothesis, not the background knowledge. And so that is where the Duhem-Quine thesis emerges, which is that you can't falsify a single hypothesis in isolation because predictions rely on somewhat of a web of assumptions, right? And so in practice, scientists can make a convention to blame the hypothesis that's under test, right? Unless, again, there's good reason to suspect the data or the auxiliaries, but no one actually knows if it's actually the hypothesis that's at fault. It could also be one of these background assumptions. So I hope that's clear here. So how does this all relate to cladograms in the study of phylogeny? Let's now apply this particular criteria to cladograms, right? And so an important point here is that Popper's his criteria of falsifiability, it refers to the logical possibility of a hypothesis being refuted, not a guarantee of an actual refutation in practice, right? This is something that's really important. And so there's a difference between falsifiability and falsification. And what do I mean by that, right? Methodological falsifiability means that the hypothesis, at least in principle, rules out something, right? That's a must. It has to rule out something. Now, even if we can't actually encounter that particular refuting case, at least at the very least, it's in principle, it's making it out to be that case, right? So even if we never actually end up observing non-E, it must predict some non-E, right? Now that's different to what's known as empirical falsification, which is that empirical actual falsification might be unachievable due to the fallibility of observations as well as the Duhem-Quine problem. So it's okay for not having an actual empirical falsification. We must have, at the very least, a methodological falsifiability in place, right? So Popper accepted that like we can never have perfect knowledge of our background assumptions or even perfect data. Testing is all provisional in that sense anyways. But the point here is that the hypothesis must be logically falsifiable to be scientific, and it's about the hypothesis form, not whether it has actually been falsified, right? So in other words, it has to qualify and it has to pass that the modus tollens kind of framework even if we don't actually find not E or that E event, that impossibility of E, that's not really what matters. What matters is that at least it predicts some kind of not E event. And again, I repeated this point, but I think it's really important, which is that the more a hypothesis forbids something, or in other words, the more ways that a hypothesis could be falsified, then the greater its empirical content and the greater its explanatory power, right? So if a theory survives a kind of harsh test that could easily refute or falsify it, then we have very strong confidence in that, right? So this is how we understand corroboration, for example, right? The degree of corroboration of a hypothesis kind of earns or increases depending on the severity of the test, right? So if the test is very hard, then the corroboration is even better, right? Whereas if the test is very light, or even if it's a non-falsifiable test, then there is no corroboration. This is important to understand because we'll see later on that one of the strategies that these evolutionary biologists come up with is that they say that we can have corroboration without falsification. So essentially what they're saying is that they would concede, they say, look, maybe it's true, maybe cladograms can be falsified, but at the very least they could be corroborated. And we will look at that as a potential solution and whether or not that works later on, right? But the idea here is that at least in Popper's framework, corroboration is only meaningful if the hypothesis was falsifiable to begin with. if there is no conceivable test that could refute the hypothesis, then saying that it's supported by evidence is really, it means nothing, right? It's somewhat of an empty claim. Now, like once we apply this criteria to phylogenetics and to systematics, the question becomes, again, are cladograms falsifiable? And I hope you see, at least from the example that I gave previously, that cladograms obviously fail logical falsifiability tests, right? We can always redistribute those traits to fit any specific cladogram, right? Now, to actually answer that question in a more robust sense, we first have to answer, or we first have to at least understand that before we even worry about how to test a hypothesis empirically, we have to ensure that the hypothesis is in fact falsifiable by finding at least one potential falsifier for that particular hypothesis, right? So we need an observable statement that the hypothesis forbids. Now, when it comes to phylogenetic trees, what would actually count as a potential falsifier for a phylogenetic tree hypothesis, right? Again, if we saw in the last, if I were to go, oh, it's gone now, but what we saw was that, again, if we have some type of species A and B, and they share some type of ancestral trait C, what we showed was that we can redistribute C, which is that synaptomorphic trait, we can redistribute it in any particular way for it to match our hypothesis, right? And so we saw that we can homologize those traits, in which case we have a particular tree, or we can redistribute these traits in a way that's kind of homoplazious, right? In a way that is independent of, or that trait is independent of common ancestry, right? And so what that means is, again, for any candidate that falsifies the observation, or for any observation that can falsify the tree will always accommodate by having this ad hoc extra change that it puts into it, right? And so we cannot say, for example, species X and Y is truly a clade, because the reason why we can't say that is because if that hypothesis were true, it's like saying if species X and Y is a true clade, for example, then it will never observe Z, right? We can't make statements like that because there is no such Z that evolutionary theory won't allow into the framework somehow. And so I hope I'm making sense here, which is that cladograms, strictly speaking, they fail that criteria of falsifiability. So now, one of the many systematic tests that exist out there, right? They test how good a particular cladogram is, and they do this by using what's known as the congruence test, right? Again, anyone who studied biology or who studied phylogenetic trees and systematics knows what the congruence test is. But the question is, does the congruence test actually disprove what I'm saying? Does the congruence test falsify phylogenetic trees? And essentially, in cladistics or in systematics, the congruence test tries to check if different characteristics or character traits, whether those be genes or whatever it may be, it tests to see if, at the very least, it tells a consistent evolutionary story. So if a tree maximizes congruence or agreement among characters, then that tree in particular is preferred, right? Parsimony analysis is a type of congruence test. And then on the next slide, I'll go over what these parsimonious analyses end up invoking and how they work. But the way it works is essentially in practice, parsimony analysis or parsimonious analyses, it's a type of congruence test in the sense that it tries to find the tree where the characters are the most consistent. Again, by that I mean they require the fewest contradictions, or they require the fewest ad hoc assumptions, or in other words, they invoke the fewest amounts of homoplazious events, right? Now, the thing is, you can even form the congruence test through somewhat of a hypothetical deductive model of testing, right? So again, I explained the hypothetical deductive model on the previous slide, but we can even show the congruence test through that model of testing where the hypothesis being tested is in fact the cladogram, right? So the tree typology, and the background knowledge is standard evolutionary biology. And then the observational statements are basic statements, right? And those are the kind of the character evidence that's interpreted as synaptomorphies. And so if a character, so let's say for example a DNA site or a morphological trait, if that particular character doesn't fit the tree or it requires too many extra changes, then that would be seen as a failed prediction for the tree hypothesis, right? So then under this view, a tree with lots of homoplasy is one that has been effectively falsified multiple times by different characters, whereas a tree with minimal homoplasy has withstood attempts at falsification, right? Is this really a solution to the potential falsity of tree hypotheses? And if you actually think about it, it's actually not a good solution, right? And so this is because the congruence test is not really a direct test of the tree hypothesis, but rather it's a test of the character hypotheses, right? So it's testing not the tree itself, but it's testing the character hypotheses themselves, right? The character traits. So each character is first viewed as an independent hypothesis of homology, for example. So, for example, trait X in species 1 and species 2 is a synapomorphy from their common ancestor, right? And when we do cladistic analyses, we are basically checking which of those character hypotheses can coexist consistently on the same tree. And if a character is highly incongruent, then we often conclude that the character hypothesis was wrong. That particular character hypothesis was wrong in the sense that there wasn't a true homology, rather it was homoplasy, right? And based off of that, we would rule it out or we would exclude it. Now, do we usually, rather, the thing is that we actually don't throw out the tree itself when we're doing this. All we do is we just reinterpret or we adjust the interpretation of that particular character. So we just change its distribution, right? We don't actually throw the tree out itself. And so the tree that emerges is one that maximizes the number of accepted homology hypotheses, right? Or synapomorphies, and minimizes assumed homoplasies, right? So the falsification is really happening at the level of the character hypothesis and not at the level of the tree hypothesis. And so the tree itself is selected to somewhat best accommodate the characters rather than being outright falsified by them. In other words, the tree hypothesis never made a clear prediction that a particular character must be synapomorphy or at the very least not a homology or not a homoplasy, right? Because it doesn't make that prediction, it doesn't falsify the actual hypothesis. And so, for Popperian hypothesis or for Popperian falsification to work, the hypothesis and the background knowledge, it needs to imply something like if this tree is correct, then character A cannot evolve twice independently. But again, evolutionary theory never gives us that guarantee because convergence and homoplasious events are always possible no matter what observation we're looking at. And so a cladogram does not really forbid character A appearing in unrelated taxa, so to speak, right? It only implies that if that does happen, then all we do is we have to label it as homoplasy. We don't actually falsify or rule it out. So saying that these species share trait X due to common ancestry, that isn't really an observable claim. The observable part is just they share trait X, for example. And that could be true. Maybe they do share trait X, but it doesn't really falsify anything, right? Because the rest of the hypothesis, which is that because they share character X, therefore they are derived through homology, that itself is theoretical, and that's not a direct observable observation. And so if they share X, but the tree says that they shouldn't, then we just say, you know what, that's just homoplasy. And again, it doesn't really falsify anything. And so I hope you guys see because of this that no cladogram is rejected unless there is a better cladogram that fits the data more parsimoniously. And again, it's not a matter of comparative fit, right? It doesn't really do anything to actually falsify the tree itself. And so therefore, these congruence tests, they look at characteristics, they do not look at cladograms themselves. Now, I have an example of this here, right? So here we have three particular hypotheses, right? What these cladograms are doing is that they're showing three different hypotheses of how humans, chimps, gorillas, and orangutans are all related to each other. So, for example, in this hypothesis, what it's showing is that humans and chimps are closely related, and then gorillas are more distant related, and then orangutans are the most distant related. And this one, what it's showing is that humans and chimps, again, are also closely related. What it's showing is that humans and gorillas are related, right? And between humans and gorillas, chimps and gorillas are even more closely related, and then orangutans are the furthest apart. And the last hypothesis, what it's showing is that humans and gorillas are more closely related, and then humans and chimpanzees are more distant related, and humans and orangutans are even further. They're even further. Sorry, they are even further related. So then the question is, okay, how do we know which one is true? How does this congruence test actually works. So again, what the congruence test is doing is that it's saying we have to find the most parsimonious hypothesis, right? In other words, it's asking we have to find the one that has the least amount of ad hoc homologies, right? Oh, sorry, the least amount of ad hoc homoplasies, right? So what we would do is we would sequence their genomes, for example, right? So these are a particular gene segment for the humans, the chimps, the gorillas, and the orangutans, right? And we want to look for what's known as informative site. So an informative site is basically places where where their DNA are matching, right? So for example, in the case here where they're all C's, right? This would not be a good place to start because this doesn't tell us anything. Similarly, A doesn't really tell us anything. Also, places where none of them are the same. So, for example, G, 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 and T, right? Again, this doesn't really tell us anything. So there's some more examples, right? But what does tell us a lot are these informative sites. So the informative sites are places in the genome that are shared by some and not shared by others, right? And in particular, they're shared by two organisms. So in this case, humans and chimps, right? They share this particular segment in their DNA, right? And gorillas and orangutans, they share this particular segment. Now we can use this information, right? And we can plot this onto these trees, right? That's how the congruence test works, right? Let's take G's, right? So what it's saying is that humans and chimps, they both share a G. So in this case, I would put this as over here as a synaptomorphic event, right? So this is G, right? So in this example, this would be G. So this particular trait G is shared by both humans and chimps. At least that's how G looks on this particular cladogram. Whereas T is shared between gorilla and orangutans, right? And so we would say that between gorillas and orangutans, we would say that they show up here. Now, what this is important because this is saying that this trait T is not shared by humans and chimps. That's why it's only applies to G and O, in the sense that both gorillas and orangutans share this tree event, this T gene, right? Whereas humans and chimps share this G event, right? Or this G sequence. Now, let's do the same, right? For this case, let's say that orange is going to be G and let's say that red is going to be T, right? On this particular cladogram or this hypothesis, how would we write this? So chimps and humans, again, share G, right? So I'm going to go ahead and put chimps and humans as sharing G, and then the gorilla and orangutan are going to share T. So I'm going to put them here. Notice I on this model, I did not put the red here, right? I did not put it here because that would imply that it's shared between both G and C, right? Or both chimp and gorilla, but it's not, right? That would be misleading. It should belong here because it's only shared between gorillas and orangutans. Now, for the third hypothesis, how would this look, right? So we have again humans in gorilla, and so what we would say is that gorillas have a T event, right? And then humans have this G, and then gorillas have this T. And likewise, the chimp also has a G, and then the orangutan will have a T, right? I know this is somewhat confusing, but I hope this kind of makes sense, right? So what a congruence test is doing is it's looking at which one is more parsimonious, which one is simpler, essentially, right? And so what it's doing is it's saying, look, in terms of this, there's going to be four homoplasious events, right? In this one, there are four homoplasious events. Once again, here there are four homoplasious events, right? But here there's only two homoplasious events, and there is one homologous event, right? What that means is that this particular model or this particular hypothesis invokes the fewest explanation or invokes the fewest amount of ad hoc explanations, right? So hypothesis 2 has four ad hoc assumptions, hypothesis 3 has again four ad hoc assumptions, but hypothesis 2 only has two ad hoc assumptions and one homologous assumption. Therefore, based off of the congruence test and based off of parsimony, this hypothesis wins. But so again, that's how the congruence test works. But again, the issue is that we're not really testing the tree itself. If you notice closely, we're not testing if the tree itself is actually reliable. What we're actually testing is how do these character traits fit into the tree itself, right? So we could have had multiple different ways of distributing these events or these traits, but the point is we're not testing or we're not concerned about falsifying the tree. We're concerned about distributing and finding the simplest tree. And so that's why this doesn't really pass the principle of falsification because it's not really testing the tree. And the trees are will always be logically consistent with whatever distribution pattern we're going with. So that's not really the way to falsify a particular diagram. That's why this doesn't pass Popper's falsification criteria. So now what we're going to be doing in this part of the presentation is we are going to go ahead and show the different strategies that multiple evolutionary biologists attempt to do in order to save this entire field of phylogenetics and in order to show that these trees are in fact falsifiable, right? So that they do become scientific. So the first strategy is that essentially, okay, before I even mention these strategies, let me first talk about something really important, right? So this is the mindset that these systematists have, right? In the parsimony camp that we saw, right, they argued that even if falsification fails, we can still apply Popper's methodology by avoiding ad hoc assumptions, right, or ad hoc hypotheses. Now, what these people would do is that they would say that Popper himself recommended that scientists reject ad hoc modifications by merely try to, or sorry, Popper said himself that we should reject ad hoc modifications that merely try to save a theory without adding new testable content. So in phylogenetics, this idea was basically we should prefer the tree that minimizes ad hoc explanations or homoplasies, right? And these people who invoke parsimony as a way to falsify the particular tree, and we showed why that doesn't really falsify the tree. But what these people are basically doing is that they're saying, or they're reinterpreting Popper to say that Popper's, or at least parsimony, does hold true for Popper's justification, in the sense that the shortest tree or the fewest evolutionary changes introduces the fewest assumptions, right? And so therefore it's the least ad hoc and therefore the most preferable. Now, again, is this really a viable strategy, right? And so I explained it before, it's not really a viable strategy, but then there's some more stuff to say about this, right? Now, if we understand what Popper meant by ad hoc, we would understand that this is actually not a good strategy, right? So, according to Popper, an ad hoc hypothesis is one that it's added solely to accommodate some type of troublesome fact, right? So it's kind of like an scapegoat. That's what an ad hoc assumption or ad hoc explanation is, right? And the most important part is that for Popper, ad hoc assumptions reduce the falsifiability of the particular hypothesis, right? So, for example, if your theory says that all swans are white, and then you find, for example, a black swan, then you might add somewhat of an ad hoc phrase on top of it, and you're going to say something like, all swans are white unless they live in New Zealand, right? So now you found an observation, and what you did is you tweaked your hypothesis to make it somewhat ad hoc in order to save the hypothesis, right? So again, the initial hypothesis was that all swans are white, and once you found a black swan, you tweak the hypothesis to say that all swans are white unless they live in New Zealand, right? And so again, this saves the theory, but makes it less testable, right? Because now you have to also specify location as well. So Popper allowed those kinds of auxiliary hypotheses that do not decrease falsifiability. You can add an assumption if it leads to new predictions or tests that themselves are also falsifiable, but you can't do that just to escape falsification, right? Or to escape refutation, right? And so that's why parsimony does not really work here. So essentially invoking Popper's no ad hoc rule doesn't really solve the problem for cladograms, right? And basically some homoplazious hypothesis in a cladogram, it's not ad hoc the way that Popper is using it, right? Because again, Popper is using it to save the falsifiability. He's saying that we can't have ad hoc assumptions that are used to save the falsification of something, right? But what this strategy is saying is that we're using ad hoc, or we're minimizing ad hoc explanations, but they're equivocating on what they mean by ad hoc, because for them, ad hoc is not really something that saves the falsification, because the falsification is always already at zero. Instead, for them, ad hoc is essentially something that does occur in nature. It actually does exist, which is homoplasy. Homoplasy is not ad hoc the way that Popper is using it. And again, I hope that makes sense, because what I'm trying to say is that Popper is saying that ad hoc is something that saves the prediction from failure. But the way that these systematists and phylogenetic and those who are studying cladograms, the way that they do that, the way that they're invoking ad hoc assumptions, they're saying that we're minimizing them, but those ad hoc assumptions are not falsifying the criteria, because the criteria is already non-falsifiable. Those are actually genuine experiences that happen in modern biology, right? Homoplasia is a genuine thing. It doesn't really save the hypothesis. So again, that's why the strategy fails. With regards to the second strategy, right? The second strategy says that the Popper kind of allowed test or treatment of probabilistic hypotheses to pass the falsifiability test, right? So what I mean by this, Popper recognized that statements like, for example, the probability of tossing heads is 0.5, right? Now, Popper says that statements like those, they cannot be falsified by a single toss, right? Because one heads or one tails doesn't really refute it. And so in fact, there's no finite set of observations that could strictly falsify a probability hypothesis. And that's again because there's always a chance for any outcome to occur, right? So what's Popper's approach to this was that we should allow a concept of being practically falsified, which is not the same thing as being falsified, but almost falsified or practically falsified, if the observed frequencies deviate significantly from the expected probability, right? Essentially, you would reject a probabilistic hypothesis if the results are very improbable under it, even though logically it's not really impossible. Some argue that cladograms are very much like these probabilistic hypotheses, right? So, for example, a maximum likelihood tree gives a likelihood score, which is probability on the data given the tree and a model, right? They're saying that these cladograms are very much like those probabilistic hypotheses. So you can basically say no single observation falsifies the tree. However, if the tree's likelihood is much lower than another's, or if its posterior probability is very low, then we can consider it falsified or less corroborated in a Popperian sense, right? So, in other words, what they're saying is that you can assign degree of corroboration to cladograms based on their likelihood or their statistical support, just like how we might reject a probabilistic hypothesis after enough data. So essentially, the argument goes something like if Popper allowed for degrees of falsifiability or corroboration for probability statements, then perhaps we can do the same for trees. So you can treat a tree's likelihood as an indication of how well it has survived specific tests, even if it never truly falsifies, or even if those tests never truly falsify it, right? But then there's two major issues with this strategy, right? And the first is that Popper's view on testing probability is actually somewhat controversial, and many philosophers even criticized him during his own time, during his own formulation of it, right? So Popper's own approach to probability, you can somewhat disregard that as being fundamental to his hypothetical deductive model, because it doesn't really add much to it. It's already on shaky grounds, right? But more importantly, and this is like the second issue with it, with the strategy, and it's a lot more sophisticated, which is to say that cladograms are not actually probabilistic hypotheses in the way that Popper was using them anyways, right? Again, even if we accept Popper's assessment of probabilistic hypotheses, modern cladograms are not really giving us those kinds of probabilistic hypotheses anyways. Popper's hypotheses are statements like whenever condition B, event A occurs with probability R, for example, right? So they're like if condition B and event A occurs with probability R, given that B, right? So for example, the probability of a P being green, given this particular Mendelian cross, is three fourths, right? So here the value 0.75 is part of the hypothesis and can be tested with enough scientific trials, right? We can crossbreed multiple plants, grow peas, and if they're green 3/4 or 75% of the time, then we can incorporate that into our hypothesis testing, right? And so, in such a hypothesis, the probability of what is being asserted is quite fundamental, right? So if we compare that to cladograms, cladograms don't really give us that kind of probability where we can observe any particular trait distribution, right? It's a deterministic statement about relationships, and the likelihood or posterior probability we calculate after the fact is not part of the hypothesis itself, right? So saying something like tree X is the true evolutionary tree, right? That we can't actually, we have no probability based hypothesis to test that, even if it is probabilistic, even if it is a probabilistic hypothesis. So it's basically an assessment of how well data fit into trait X under a particular model. So if that like that tree X has a low likelihood given the data, then that means that another tree might fit the data better. But again, tree X itself didn't predict a specific probability distribution that is now falsified, right? We're just comparing models. So the tree hypothesis doesn't really say something like I expect data set D with probability 0.9, for example. It doesn't say anything of that sort at all, right? So therefore, you can't falsify a tree by saying its likelihood is low because a tree never promised a high likelihood to begin with. And this is why strategy two fails because it kind of misidentifies the nature of hypotheses, right? And what probability is, right? You can't magically give cladograms a falsifiability by borrowing it from statistical models. Even if one tree has a higher likelihood, both trees remain unfalsifiable. Both trees remain unfalsifiable kind of logical structures. We're just measuring what fits the tree better, but we're not actually measuring the falsifiability of the tree itself. You can use maybe something like likelihood or Bayesian posterior to kind of understand these traits better. But again, they don't really falsify anything. They don't change the non-zero falsifiability of cladograms themselves. And this is actually quite important because there's a lot of literature out there. There's a lot of papers that test different phylogenetic models. There are multiple software algorithm run or algorithm based computations that try to show which one is more likely and which one's less likely. But again, it's not really falsifying anything here. And so I hope that kind of makes sense here. So to summarize what strategy two is saying, they're saying that we can't falsify a cladogram with a single trait, but what if we treat it as a statistical hypothesis, right? As we accumulate data more and more trials end up accumulating data, a tree that consistently has low likelihood or requires many ad hoc assumptions could be considered practically falsified. But again, the response to this is that cladograms aren't formulated as statistical hypotheses that predict probabilities of outcome. So that's why this analogy kind of fails. You're just measuring what fits the tree best. You're not actually subjecting the tree to a Popperian test, right? So a tree doesn't predict the exact data that you got with a quantifiable probability. It's the evolutionary model that does that. So like that particular computation that's doing that. So you're not really falsifying the tree. You're just choosing a better supported model. And so that might be scientifically valid. I'm not saying that's not scientifically valid, but it's not justifiable, and that's the whole point of this. And so lastly, like the third kind of strategy is basically it's basically invoked by some people who they would concede and they would say maybe cladograms aren't falsifiable, right? But can't we still say that even if they aren't falsifiable, aren't they still to some extent supported or corroborated? Can't we basically decouple corroboration from falsifiability? And again, to understand this better, remember when I said that for Popper, corroboration implies falsifiability, right? You can't decouple them. But what this strategy is trying to do is it's saying we can actually corroborate a particular tree without having the need to falsify them. Now, if we go down this route, there's a major issue. Given Popper's logic, because corroboration is linked to tests that could have falsified the hypothesis, if no such test exists, you can't really say that the hypothesis is corroborated by anything, right? So any support that you feel it does have, it's illegitimate or even metaphysical, right? It's not an actual testable observation, right? Again, it's like pseudoscience. It's like saying, I feel good about something like, I don't know, my horoscope predictions, right? But just because you're feeling good about your horoscope predictions doesn't mean that this whole field of astrology is actually scientific or it's scientifically corroborated, right? In other words, just because something fits with the data better, right? that doesn't really imply that therefore it's of any use, right? So if basically what I'm saying is that if you attempt to assign degrees of support to cladograms that aren't falsifiable, then you're not really addressing the main issue at hand, right? Which is that even if you have some kind of consistent narrative in the phylogenetic tree, you're still not having a particular hypothesis that could be or that could be falsified. You're not actually willing to reject that hypothesis given data. And that's like where the whole issue is coming in from. So to conclude this entire thing, the main point again in this whole presentation is that if we stick to Popper's falsificationism, then neither parsimony or likelihood provides a somewhat truly scientific justification for choosing one cladogram over another, right? And that's because in the end, they're not testing and falsifying a tree hypothesis, but they might be testing other things, right? So, for example, parsimony and likelihood, they're testing maybe goodness of fit criteria. So, for example, they're testing simplicity or explicit model probability, but neither is actually performing Popperian hypotheses refutation themselves. They're not actually falsifying the tree itself. And so they are picking the tree that best fits the data rather than eliminating the tree by contradiction. And so Popper's model is basically it's under the guise of observable traits. What this means is that phylogeneticists have to start developing their own philosophy that kind of acknowledges these particular realities. They can't really rely on Popper, and the whole key point in all this entire presentation is that these phylogenetic trees are not falsifiable. And this again is it's going back to the conversations that I've had or the lectures that I've given on the testability of universal common descent. And that is, again, I think episode 3 of this series. But I think this is a good conclusion for this particular segment of the series, which is that these phylogenetic trees do not give us sufficient evidence. They're not testifiable in a particular way that could falsify this entire endeavor.